Wonderful. Welcome, everyone. On behalf of Muscular Dystrophy Canada, welcome to our Moving Research Forward, the results of Muscular Dystrophy Canada's 2024-25 Research Grant Competition. My name is Hamera Osman. I'm the VP of Research and Public Policy at Muscular Dystrophy Canada. Today's announcement is available in French via the Wordly app, which is embedded right now into your Zoom platform. If you'd like to view today's announcement in French, there will be a link that will be posted in our chat in just a few moments, and you'll be able to click on that link and receive today's information in French. So really excited today to provide you an introduction to the grants that we're supporting this year, thanks to your support. All of the grants that we're supporting today really aligns with our strategic priority, strategic plan. We're in year 3 of our strategic plan, really with our strong commitment to research and breaking down barriers. And you're going to hear a lot more about this throughout the webinar today. We have been able in the past year invest in research, not only in the research grants that you'll be hearing about today, but we have also been able to support 2 clinical fellowships. We've been able to fund 3 postdoctoral fellowships, and 355 hours were dedicated to review the grants that we're talking about today, as well as to support webinars like this and the Neuromuscular Disease Network for Canada. These are clinicians, academics, researchers who are volunteering their time to support our research initiative. So we're really grateful for their support. And at MDC, not only do we provide funding support for research grants, we also run research grants ourselves, research projects ourselves. And just in the last year alone, we have over 1,800 participants in research projects. Many of you on today's webinar have participated in journey mapping initiatives, in health experience, or in our burden of inherited neuromuscular disease study. So really, as many of you might know, Muscular Dystrophy Canada is a health charity that is supported by individuals, by donors, by a community, by Canadian firefighters, by clinicians, by scientists. And really what we're doing is turning that generosity, those donor dollars, into action. When we think about our research strategy, we're investing in the projects that you're going to hear about today, but also people like those fellows. Postdoctoral fellows are individuals who've completed their PhD training and are now doing specialized training in laboratories like the ones that you're going to hear from to specialize in a different research project. We also, when supporting grants like this, are supporting people such as not only the trainees, it might be lab managers, it might be technicians who are able to carry out the work. We're also able to really leverage those gifts, those supports that you provide us in turning into investing into partnerships, influencing public policy. And I did want to take a quick moment to share how we even came to the 9 highest awarded grants today. We had a call for clinical and translational projects back in September. We received 28 applications. As I mentioned, many reviewers were involved. We also had community reviewers. These are individuals with lived experience. They're either personally impacted by a neuromuscular disease or have a family member. And they provided impact on the relevance, the types of questions that were being answered, and their feedback went into today's consideration for the top ranked awards. So I'll pause there and I'll go ahead and turn it over to you, Dr. Parks, as you'll share more on the value of research. As I see that's coming up, Dr. Parks, I should once again just formally thank you for supporting our research grant competition. Dr. Parks chaired the Translational Science Research Grant review process, so we really appreciate that. And he is the chair of our Medical and Scientific Advisory Committee. Over to you, Dr. Parks.
Great. Thank you, Hamera. So it's a pleasure to be able to talk to you today a little bit about Muscular Dystrophy Canada and research efforts. So as you likely know, Muscular Dystrophy Canada supports people affected by over 150 neuromuscular disorders. And for several of these disorders, such as spinal muscular atrophy and Duchenne muscular dystrophy, we know what the disease-causing gene is, and we know quite a bit about the biology. We don't know everything, but we know quite a bit. But for many of these other disorders, there's still many things that need to be uncovered, such as what is the disease-causing gene and how does it give rise to the deficiencies? We need to develop and to test new therapeutics in animal models of these diseases. We need to determine the natural progression of the disease in affected individuals so we can keep a better track of the progression of the disease and inform these individuals what they will come in the future. And we also need to develop best practices for managing disease symptoms in these infected individuals. And there's many other things that we still need to discover. And this is why Muscular Dystrophy Canada invests in research. And there's two types of research that it is involved in. The first is the Translational Science Seed grants, which are designed to for the development of potential diagnostics or therapeutic interventions using cell and animal models of neuromuscular disease. So these are really the lab-based studies. So it's individual scientists or clinicians working in a lab trying to come up with answers. The second type of grant is the clinical research grants, which are designed to research to improve treatment, clinical care, or quality of life in individuals affected by neuromuscular diseases. And these are really the patient-based studies. And it's success in these two types of research that really leads to new treatments. And the example I'm going to give is the timeline for development of nusinersen or Spinraza for the treatment of spinal muscular atrophy. So the clinical disease was really first identified in 1891, and it wasn't for 100 years before they came up with the causative gene. So once they had identified the gene, we could then do lab-based studies to try and understand how mutations in that gene gave rise to the disease. We could develop animal models to mimic the disease, so they're very similar to what happens in the human condition, and we could start developing therapeutics, which could then move into the clinic and start to be tested in patients. And in 2016, of course, the first approval of nusinersen became available. So there's two things I want you to see from this timeline, this brief timeline I've shown you. First is research success takes time. So although the gene was identified in 1995, it wasn't until 20 years later that we came up with the first therapeutic that was available to patients. So sometimes it takes longer, sometimes it's a little bit quicker, but it does take time to develop these therapies and understand the mechanism of these diseases. The second thing I want to point out is, although I've shown you 5 or 6 sort of milestones along the development of this therapy, really it's hundreds, if not thousands, of individuals and individual research projects that really gave rise to this success. So it's not one individual working in one lab that came up with all of these discoveries. It's hundreds, if not thousands, of individuals and individual research projects that gave rise to the success. So the idea now is to try to apply this type of model to other types of neuromuscular diseases. So Friedreich's ataxia, LPMD, FSHD, some of these other diseases that are less well characterized and perhaps don't necessarily have effective therapeutics. The idea is to do research to try and understand these disorders a little bit better and come up with appropriate treatments. So how does MDC decide what grants to fund? Well, it's a process called peer review. So scientists and clinicians come up with ideas of research that they think will help contribute to the neuromuscular community, and they submit grants to Muscular Dystrophy Canada. And the grants are reviewed by an expert panel of 6 to 8 scientists, clinician researchers, and as well as lived experience readers to critically evaluate the grant's goals, strengths, weaknesses, and potential impact. The applications that receive the highest scores that are deemed highly meritorious and likely have significant impact in the neuromuscular disease community are supported for funding. Also importantly, the unsuccessful applications are given feedback, and the idea is to inform these researchers of where perhaps we think that they could improve their grant application and make it more amenable to the research community, and submit again next year and possibly be successful in that competition. So what happens after the MDC awards the grant? Well, over 1 to 2 years, the investigator completes the goals of the grant. So what they put down on the piece of paper, they try to complete those goals. They move the science forward, they generate data and advance our knowledge and drive progress and innovation. What's also important, as pointed out by Humira, these same funds support the next generation of neuromuscular disease researchers. So this is the graduate students, the postdoctoral researchers, other research and clinical personnel. So the next generation of researchers in neuromuscular disease. At the end of the granting period, a final progress report must be submitted to MDC to help track the impact of the MDC-funded research. And MDC also coordinates with investigators to ensure knowledge gained from these studies is relayed to the community to maximize impact. And these are the webinars and the publications that you see come out from Muscular Dystrophy Canada. What's also important is that these seed funds that Muscular Dystrophy Canada gives to support this research can then be leveraged to apply to other organizations to get more money to fund future research in neuromuscular disease. So these are grant applications that go to agencies like Canadian Institutes of Health Research, which can really move the research forward. So just in summary, MDC supports research and training. They hold the researchers accountable to make sure that the goals of the grant have been achieved, and they make sure that the end users, the people who are impacted by these neuromuscular disorders, receive the information that's gleaned from these studies. And they also, through these studies, invest in future research in neuromuscular disease. And with that, thank you.
Thank you very much, Dr. Parks. I'm just going to go ahead and share my screen again, which brings us to say, after a really thorough, comprehensive peer review process, we're really thrilled that this year we are now funding 9 different research projects that are translational and clinical projects. So congratulations once again to the recipients and to your teams. I did also want to take a moment before hearing from each awardee that this year we did run what's called a sub-call. So a sub-call is where we were particularly looking to support 2 projects on a condition called Friedreich's ataxia. As you know, at Muscular Dystrophy Canada, as Dr. Parks demonstrated on that one slide, we cover hundreds of neuromuscular conditions. And it's really important in certain times to really have a dedicated call to attract research projects on, in this case, a rare neuromuscular condition. So within this call, we're really grateful also to have Dr. Yokota and a member of Dr. Cynthia Gagnon's lab to speak about these 2 projects. With that, I am really thrilled to again have each of these recipients talk about their research projects. And I will pass it over initially to our Translational Science Seed Grant recipients, starting off with Dr. Burrell. I'll go ahead and stop sharing my screen.
Okay, do you guys see my screen?
Looks good. Go ahead.
Good. All right. Well, first of all, I would like to thank the association for awarding me this grant. It's a great honor, so I'll try my best to deliver. So the title of this grant is Targeting Mitochondrial Quality Control to Promote Muscle Regeneration in Duchenne Muscular Dystrophy. So basically, skeletal muscles are endowed with a unique ability to repair, and this is by virtue of their resident muscle stem cells. So this is illustrated here, this immense capacity to regenerate muscle fibers. We see muscle stem cells that reside on top of mature muscle fibers, and those cells, upon injury, can exit their normal dormant state, activate, divide, multiply, basically, and to form new muscle fibers. And this can happen repeatedly for efficient muscle repair. And you also see that a certain proportion of these cells are able to undergo a process called self-renewal to replenish the muscle stem cell pool, just to ensure that they're able to trigger repair repeatedly over the life course. And what we know in Duchenne muscular dystrophy is that these muscle stem cells do not behave properly. They don't actually self-renew as easily, which can lead ultimately to stem cell exhaustion and also impaired ability to repair muscle on the long run, which is an issue in this disease. And so what we're trying to do is to understand why is that, and whether we can target these malfunction with drugs to improve muscle regeneration. So in the past few years, our lab and others also have shown that the ability of muscle stem cells to mount an efficient repair and self-renewal response is dependent on the efficiency of their internal powerhouses, which are called mitochondria. Those mitochondria are essential for proper cellular function as they generate energy and also a variety of signals that regulate how cells behave when facing certain circumstances. In muscle stem cells, which we have shown with experiments, is that if we interfere with mitochondria, either by poisoning their power plant, if you will, or disrupting the natural process by which they're maintained or recycle. This process is called mitophagy. What you end up having is a loss of stem cell ability to self-renew, and you end up with an impaired muscle repair capacity. And more recently, what we found is that mitochondria mitochondrial function and mitophagy are disrupted in muscle stem cells from Duchenne muscular dystrophy mice. So more specifically, mitochondria show signs of altered energetic function and also oxidative stress. In addition, we also find that mitophagy, which this recycling process, is also impaired, and that we think that this contributes to the buildup of malfunctioning mitochondria, and ultimately this results in an abnormal muscle repair response. And importantly, a compound known as urolithin A, which acts in part by stimulating mitophagy, has shown encouraging results in DMD mouse models. So urolithin A was shown to improve muscle repair abilities, resulting in better preservation of muscle force, and also increased survival in this mouse model of DMD. So in a nutshell, the project that we have is basically to determine, using healthy and MDX mice, whether and how mitophagy and mitochondrial quality control becomes deficient in the MDX mouse model, how this impacts muscle stem cell function, and also their ability to repair, regenerate muscle upon injury, as well as beneficially impacting the ability of muscle stem cells to regenerate tissue when they're transplanted in a stem cell therapy. So this is the first of the two objectives, and the second one is actually to determine the therapeutic potential of 4 different mitophagy stimulating compounds. The first one is urolithin A, for which we already have shown to have beneficial therapeutic potential, and also 3 novel compounds that are more potent than urolithin A and or more specific. And so we're hoping to even increase the therapeutic potential even more. So overall, if I want to give you an overview of the potential importance of this proposal, in DMD, gene replacement therapy basically represents the holy grail for the treatment of the disease. However, for the moment, these approaches have major limitations. They benefit only to a subset of patients, and critically, they do not address the progressive exhaustion of muscle regenerative capacity. So our research will provide a comprehensive analysis of mitochondrial quality control, how it functions, and so we will be able to understand how this can lead to loss of regenerative ability in DMD. And most importantly, from a therapeutic perspective, we'll provide data on the therapeutic potential of novel mitophagy stimulating compounds with increased potency and specificity. And for some of these compounds, the regulatory path to the clinic is simpler because some of them have already been used in humans or are classified as natural products, which makes it a bit easier. So this is it. I'm done.
That's great. Thank you again, and congratulations, Dr. Burrell from the University of Ottawa. Up next, we're going to hear another rapid flash talk on the supported research, which is from Dr. Mark Curry from the University of Toronto. So I'll go ahead and ask Dr. Curry to share his screen.
And can you see that?
Yes.
Looks good.
Perfect.
Okay, great. So I'd like to start off by extending a big thank you on behalf of myself and my co-investigator, Dr. Antonis Tatarakis, to Muscular Dystrophy Canada and its donors for their generous support of our project. So during the pandemic, we witnessed the importance of getting answers quickly. so that we could make informed decisions about vital public health matters. And PCR tests were the first diagnostic tool that was available to us, which took days and sometimes weeks, requiring a trained medical professional to acquire a sample, expert laboratory technicians to conduct assays, and expensive laboratory instruments. Now, rapid COVID-19 tests eliminated the need for expertise and fancy equipment. They empowered individuals to get clear answers at home in minutes by following a few simple instructions. So this development marked a turning point in the fight against COVID-19, leading to quicker identification of infections. So in our project, we aim to do something similar for scientists. We will create a tool that will give researchers that are studying Emery-Dreifuss muscular dystrophy faster answers. So our tool will help figure out what's going wrong in the cells and which drugs might fix it. So Emery-Dreifuss muscular dystrophy is caused by mutations in 9 different genes, all of which affect the same part of the cell that controls how genes are turned on or off. And in this process, we call gene regulation. So why is gene regulation important? Well, it turns out that almost every cell in the human body contains the same set of genetic instructions, or DNA. However, despite having the same set of instructions, each of these different cell types that I'm showing you display unique behaviors and characteristics. And this is accomplished through gene regulation. So even though these cells contain the same entire set of instructions, they only end up using part of those instructions, which is determined by which genes are turned on and which genes are turned off. It is the combination of genes that are expressed that ultimately define a cell identity. So in the case of Emery-Dreifuss muscular dystrophy, this gene regulation is messed up in muscle and heart cells, which causes a sort of cell identity crisis. These cells are not given the correct instructions they would need to make functional heart and muscle cells. So what we're doing is we're building a tool that acts like a visual alarm system by targeting Emery-Dreifuss muscular dystrophy gene regulation pathway to a reporter gene. And it will turn cells green when the gene is not expressed properly. However, if a drug or a genetic change helps fix this problem, the green color will disappear. So it's a simple system. Green means there's a problem. No green means that we've identified a possible solution. So to apply this tool to drug discovery, we will add the reporter cells to a 96-well plate. And so I'm showing here on the right, the top right of the screen, that's a side view of what the wells look like. You can see cells growing at the bottom of those wells. And so we will add different drug molecules to different wells in these plates and look for wells that lose that green color. So these wells contain drugs that could potentially be used to treat Emery-Dreifuss muscular dystrophy. Similarly, we could perform genetic screens using our tool to identify new targets for drug development. So to summarize, we will develop a fast and simple tool to monitor the incorrect gene expression associated with Emery-Dreifuss muscular dystrophy. This tool can be used for rapid drug screening as well as research that will advance our understanding of disease biology. We are starting with one of the genes that's linked to Emery-Dreifuss muscular dystrophy. However, the system can be expanded to include more associated genes to help identify treatments for more patients. Ultimately, we hope that our system will mark a turning point in Emery-Dreifuss muscular dystrophy research, much like the rapid test did during the COVID-19 pandemic. So this is a picture of my current group, and I just like to finish off by once again thanking Muscular Dystrophy Canada.
Thank you, and congratulations once again. Up next, we're really pleased to support the work of Dr. Vincent Pitcher-Martel from Laval University.
Great. So you see my screen. Okay, so hi everyone. My name is Vincent. I'm a neurologist and a researcher in Quebec City. So I just started my lab last summer, so I'm really glad for muscular dystrophy to offer me this opportunity to really get that project forward. So today I will present you my project on OPMD, which is a muscular dystrophy that we don't talk about much often. and how I'm planning to model that disease in iPSCs and muscle and 3D muscles, and with the goal of developing a new drug for them. So a short introduction on OPMD. So OPMD is a muscular dystrophy that affects mostly adults after the age of 50 years old, and present with eyelid drooping, weakness in the throat, causing trouble swallowing, and weakness in the legs that can really lead to trouble walking and the needs of using a wheelchair after a few years. So the disease is really frequent in Canada, mostly in French Canadian, in about 1% in 1,000 people in Quebec, and mostly in the Saguenay-Lac-Saint-Jean region. There's no disease-modifying therapies, unfortunately, for that disease, but we know that the disease is caused by a mutation or repeat expansion in the PABP1 gene. So when we want to study a disease, we need relevant animal models or cellular culture models. So for now, there's no really these models that really replicates what we see in human in OPMD. So we are planning to use a new approach that is really popular these days in research, which is called iPSCs and organoids or 3D muscles models. So iPSCs, or induced pluripotent stem cells, are stem cells that can be differentiated into any type of cells, human cells, and in our case, we'll develop them into muscle cells. So we can use those cells from a patient directly, or we can insert the mutation that is causing the disease into the cells, and after that, generate new muscle cells. So the plan of our project is really to generate an iPSC-derived 3D muscle that we can study and apply that model to develop a new drug. So we are hoping to create that relevant disease, hoping to generate a model that can be used also to study how the gene impact the disease and impact the muscle function. And finally, develop a therapy that we call antisense oligonucleotide, which are basically short DNA sequence that then can directly target the gene, reduce the gene or modify the gene to treat OPMD. So again, how we will do that? So we'll use iPSCs that are derived from patients carrying the disease. And we will also use iPSCs that will integrate mutation with a technology called CRISPR. And so with that, we can really model different number of repeats that can be seen in human. Those cells will be differentiated into muscle cells that are called myositis. And after that, we will generate a 3D model to see how the gene mutation affect how the muscle react, how it function, the integrity of the muscles, and how really it caused the disease. And after that, we will use that to screen some ASOs, antisense oligonucleotide, and develop a new drug. So the impact of our work will be different points. So first, we'll have a new iPSC-based models that we think that will be really relevant for OPMD, and we can share that model to other people in the research community. We will gain deeper insight of how the repeat expansion in the protein alter the muscle and how it can contribute to disease mechanism. We'll explore new therapeutics that can really affect and improve lives of thousands of Canadians. And finally, we will have kind of a pipeline for muscle therapeutic development that can really be applied to other muscle disorders as well. So shortly, I want to thank my lab and, of course, Muscular Dystrophy for that grant. That will be really useful.
Wonderful, and congratulations again. As Dr. Parks mentioned, we're really pleased to support, seed projects, initial projects like that from your lab that we can eventually build on and build out your program of research. Up next, we're really pleased to have Marie-Claude, Dr. Marie-Claude Sinsen. Over to you.
Yes, can everyone see my screen right now?
Yes.
Okay, so good evening, everyone. So first, I would like to thank Muscular Dystrophy Canada and the organizers of this webinar, as well as the donators for giving me the opportunity to present this project today. Our team studies oculopharyngeal muscular dystrophy, or OPMD, which has just been described by Vincent. So OPMD is a rare neuromuscular disease that is more frequent in the province of Quebec compared to the rest of the world. It is also most prevalent in the Saguenay-Lac-Saint-Jean region where the research will be conducted. So different muscles are affected by this disease, such as the throat, the eyelids, and the limbs. OPMD progress slowly and gradually. People affected by OPMD are limited in their movements, and they often need to use a wheelchair. So their quality of life is greatly impacted by this disease. There's presently no cure for this rare disease, and our team wants to study it to offer eventually better therapies. So OPMD is caused by mutations in a gene called PABPN1, which regulates RNA processing. So in other words, PABPN1 controls how other genes are read and turned into a function. PABPN1 is present in all cell types, which means that it is possible that all the different cells in the body could be affected by the disease. So most studies so far have been performed on muscle cells for obvious reasons, but it is totally possible also that other cells in our body are also affected by the mutation in PABPN1 and contribute to some extent to the disease state. So our research project is to discover exactly which cells in the muscle tissue present defects. We will use the most recent technologies to discover which cells are different in people affected by OPMD. We will employ techniques that are called spatial transcriptomics and spatial proteomics that were developed very recently and that allow to distinguish tissue composition and organization from patient biopsies. So we expect to find differences in muscle cells, obviously, but we want to know if other cell types, such as the satellite cells, which are also called muscle stem cells, fibroadipogenic progenitors, immune cells or blood cells, or the extracellular matrix, for example, they also present differences in OPMD muscles. So one strength of the project is that we will be able to correlate the molecular phenotype of the biopsy with clinical data from the patients, indicating disease severity, such as the age, the type of PABPN1 mutation, the muscle strength, walking speed, etc. So this project will allow us to better understand the disease and develop new therapies in the future for people who live with OPMD. So finally, I would like to mention that this project will be done in collaboration with a very experienced and multidisciplinary team. So this collaboration will facilitate and accelerate the completion of the project, and it will also open new perspectives and expand our knowledge on the molecular and cellular defects that are observed in muscles from OPMD patients, while also taking into account the clinical phenotype. So we're looking forward to launch this exciting project as soon as possible. And thank you very much for supporting us and for supporting our junior team as well.
Absolutely. We're thrilled to support your work. And I had a quick reflection as I'm listening to these talks. You know, in the past, we haven't supported as many projects on OPMD. So I'm thrilled today that we're supporting 2 projects on a condition that affects French Canadians and such powerful work. So again, really grateful to see this, and we'll be happy to see in a number of years the results of this important work. So congratulations once again. Lastly, from our translational science section, Dr. Toshi Yokota will be presenting his work. And again, congratulations, Dr. Yokota, as this is one of the Friedreich's ataxia grants that we are supporting together with Ataxia Canada. Over to you.
Yeah, thank you. Can you see my screen?
Yes, you can go.
Okay. Yeah. Thank you. Yeah, I'd like to first of all thank Muscular Dystrophy Canada and supporters for supporting our new exciting project. Today, I'd like to talk about enhanced antisense therapy for spinal bulbar muscular atrophy. And after a discussion with MDC, we also decided to start a new project, Friedreich's ataxia, using lipid nanoparticles. And and also I also have to apologize for my unavailability for Q&A session. I have next meeting in 20 minutes, so I have to leave after this presentation. And this project is collaboration between our group at the University of Alberta and several groups in Western Canada, including Dr. Pfeffer at University of Calgary and Dr. Callis at UBC and Dr. King and Dr. Schellenberg at University of Saskatchewan. So our overarching goal is to develop targeted antisense therapy for SBMA and FRDA, and we use small DNA-like molecules called antisense oligonucleotides. And especially the big challenge in antisense therapy is delivery to muscles and nervous systems. So we hope to improve the delivery system using lipid nanoparticles. And what makes our project really meaningful and unique is we partnered with indigenous communities affected by SBMA, and we hope to ensure culturally respectful research design in this project. And experimentally, we take two approaches. One is to measure effect of antisense oligos using SBMA-derived cells and FRDA-derived cells. And we look into the effect on gene expression and RNA and protein levels. And we also have a mouse model of SBMA. So we hope to measure the effect of antisense oligos in mouse models as well. So so we focus on two diseases, and one is SBMA, and which is caused by mutation in androgen receptor gene, and patients have trouble walking, talking, and swallowing, and no treatment is currently available. And antisense oligo therapy is quite a promising therapeutic approach, but delivery is challenge, as I mentioned. So we hope to block toxic gene expression using antisense oligos. And as I mentioned, we hope to improve the delivery using lipid nanoparticles, and we collaborate with UBC group. They are also involved in the development of a COVID-19 vaccine, so they are expert of lipid nanoparticle research, and we have customized lipid nanoparticles for our antisense oligonucleotides. And our hypothesis is to deliver antisense oligos in LNP, and that can work better at lower doses to treat SBMA and FRDA. So the second disease we target is FRDA, or Friedreich's ataxia, and it's a rare inherited disorder, and it causes damage to nervous system, and it typically begins in childhood and leads to impaired muscle coordination, weakness, heart complication, and it's caused by mutation in FXN gene and leading to reduced production in frataxin. So it's a loss of function disease. So in this case, we have to upregulate and restore the expression of the gene. And there are many body-wide symptoms. So in most patients, the disease is caused by mutation in intron gene, intron repeat. There are GAA repeats in intron 1 sequence, and it's increased in patients. And that leads to reduced expression of FXN messenger RNA. So we hope to use antisense oligos targeted for intron 1 gene, and that can suppress the harmful effect of the repeat, and we can restore the gene expression. And potential impact of our research, we hope to find safe and effective treatment to improve quality of life for people with SBMA and FRDA. And it can potential impact beyond just two diseases. Antisense oligos can be customized and targeted for other diseases as well. And also FDA already approved antisense oligos for SMA and Duchenne muscular dystrophy, which I was involved a little bit. And we hope to expand the applicability of antisense oligos. And we hope to identify better way to deliver antisense oligos using lipid nanoparticles. And yeah, lastly, but not least, important aspect of our research is community engagement. We partnered with indigenous communities, and our group, my student Jamie Lecky and Dr. Pfeffer and doctors in Saskatchewan also identified strikingly high prevalence of SMA observed in indigenous communities, and we are guided by community advisory circle and so to knowledge holders, and the project prioritizes culturally safe research practices, indigenous data sovereignty, and local knowledge integration. And we hope this partnership also help biomarker development and translation finding into therapies. And yeah, we hope to this research brings tangible health benefits to indigenous communities affected by SPMA and beyond. Okay, that's it from me. Thank you for listening.
Thank you, Dr. Yokota. I should emphasize that this is really exciting that we get to support work on antisense oligonucleotides in the case of FA and SBMA. As many of you might know, Dr. Yokota's research actually led to the development of a drug, vitilarsen, which is an antisense oligonucleotide, and it's approved by the FDA for Duchenne muscular dystrophy. So one of the treatments approved for Duchenne. So really hoping that there's success with the work that you're doing. So thank you again for presenting and making
Yeah, thank you.
Wonderful. So now we're going to switch over to our research grant recipients who are clinical research grant recipients. And up first is Dr. Carolina Barnett-Tapia. Dr. Barnett-Tapia. Perfect. Just gonna, oh, there we go.
Sorry. Thank you very much for giving us the opportunity to carry on this research that we hope is going to be useful for patients and clinicians. And we're shifting gears, not just from basic to translational, but disease, because I haven't seen other myasthenia gravis talks tonight. And that's the focus of our study, is multimodal remote screening for bulbar dysfunction. So I'll show you what this means because it's a mouthful. So myasthenia gravis is an autoimmune disease. Moving from hereditary diseases, there are abnormal antibodies that attack how the nerves and muscles communicate with each other. And people with myasthenia gravis present with weakness, and any muscle in the body can be affected. And this nice little diagram shows that all the muscles that are commonly affected in people with myasthenia. And if you see, we have in red fatigability marked in many of the muscles, because in myasthenia, the weakness is not fixed, is not continuously progressive, it fluctuates. So patients have good and bad days, and even in the same day, sometimes they wake up feeling very strong and have very little symptoms, but as the day goes by, the muscles become weak and some of the symptoms become more apparent. I just want to highlight, for the purposes of this study, voice and speech, meaning that patients have difficulty pronouncing words. Sometimes the voice is very soft and hoarse, and it can be hard to understand them. Then we have chewing here on the right-hand side, with jaw fatigue. So sometimes it's very mild, they just get tired at the end of the meal. But in severe cases, their entire food groups of patients are unable to chew. Like, think about something as common as steak or raw carrots. And some patients can't chew at all. And then we have swallowing, which can be as mild as a little bit of water that, goes the wrong way, and a little bit of coughing, to water or fluids coming up the nose, or inability to swallow. So we see all that range of severity for those muscles. One of the problems we have in clinical practice is that our neurological examination is not sensitive to detect weakness in the muscles that we need for speaking, for chewing, and for swallowing. Because of the fluctuations in myasthenia, often the patients don't have symptoms. So they may come to clinic and their voice sounds good, their speech sounds good, they may drink a cup of water, they're good, but they will tell you, well, you should have seen me last night or towards the end of the day. And most clinicians don't have time to do a very long assessment of speech and swallowing in a regular clinic visit. We also know that people with myasthenia gravis use very few services for speech pathology as outpatients. So compared to other neuromuscular diseases where we routinely refer patients to see speech pathologies, people with myasthenia gravis very few times actually see a speech pathologist unless they are severely affected in hospital. And we also know that actually they would benefit from those services. They can learn techniques to prevent food going the wrong way, to modulate their voice, to conserve their energy so that their voice and speech is better. So we need to find ways to identify which patients would do better with speech pathology so we can provide those services. So our goal is to develop and validate a virtual assessment platform. So the goal is that this is out of the hospital where patients can do this at home to detect their swallowing, chewing, and speaking problems. And hopefully with this can be carried to clinicians to identify which patients would benefit from actually going to see an in-person speech pathologist to have the full-blown assessment and get the care they need. So the platform already exists. We've developed with our team, and I'm not a speech pathologist, but some of my collaborators and co-investigators who are speech pathologists. It's been used for other neurological disorders such as ALS and Parkinson's disease. So these are not patients, these are some of our team, and this is done at home, so they are recording, done live. And one of the things we do is patients or individuals read a passage, and we record this, and we analyze how fast, the words per minute, how intelligible, do we understand the words, or we cannot understand them? Do they take pauses? Do they need to take a deep breath? So that's for the speaking part. In the right-hand side, this picture that looks like an 80s horror movie is actually a video. This is just a still of a video where all these little dots are actually landmarks. So we do landmark tracking, and the current software follows over 400 landmarks in the face. And we can track all the movements when patients are doing things like speaking or swallowing. And this is analyzed with the help of our biomedical engineers using AI to identify models that can help understand when these movements are normal or abnormal, or when should we start getting worried that those weakness is maybe causing some problems down the line. So we're recruiting patients, people living with myasthenia and control, because we want to see how it behaves differently. And we have data for other diseases also to compare. Everything is done virtually because myasthenia fluctuates. We will have people record twice, very early morning when they're at the best after their medications, and late in the evening when they're tired before their medication, so we can see these variations. And we'll analyze all the things I mentioned. And what we hope to have is something that's either a mobile app or a virtual platform where patients can log in, do their assessment, get a result, and that result, if there are concerns, bring it to your doctors and see if you need to have a formal referral. And with that, I want to thank, besides Muscular Dystrophy Canada, this is, as you can imagine, a lot of pieces of the puzzle, because we have my biomedical engineers, speech pathologists, we have a wonderful patient partner, and the whole team at the Prosser Family Center of Neuromuscular Disease. Thank you.
Wonderful. Congratulations again, Dr. Barnett-Tapia. We had recently done the Canadian Myasthenia Gravis Journey Mapping Initiative and really learned how much there's an impact on voice and speech and swallowing, so we welcome work like yours. Up next, we have Dr. Melissa Fascoletti. I'll go ahead and welcome you to share your screen.
Can you see my screen? Okay, great. So thank you so much for this opportunity. My project is using genes to better predict bone fragility in children with Duchenne muscular dystrophy. So youth with Duchenne muscular dystrophy are at really increased risk of fracture. And the main reasons for this is because of their weak muscles from the genetic defect in the dystrophin gene. Less muscle strength leads to less muscle activity and less walking. And so weak muscles will lead to weak bones. And in combination of that, children with Duchenne muscular dystrophy also take medications called steroids on a daily basis. And although they're extremely important to keep muscles strong as long as possible, unfortunately, they are quite toxic for the bones. So with this two combination together, what ends up happening is you get weak bones that break easily, even with mild trauma. And so this is clearly depicted here on x-rays. So the one on the right here, we see a teenage boy who has a normal knee x-ray. And here we have a teenage boy with Duchenne muscular dystrophy, and it's quite clear how much weaker the bones are. So fractures can happen in the legs, but they can also happen in the back or the spine. So as many of you know, the spine is made up of many little bones called vertebrae here. And if we zoom in, here's a nice normal vertebrae. And if we take a microscopic look, we see that the bone inside is porous, but nice and dense. And unfortunately, in Duchenne muscular dystrophy, what happens over time is that the bones become much more porous, and we call this osteoporotic bone. And just with regular daily activities, walking, sitting, the bones end up fracturing, but kind of crushing over time. And this can be, as you can imagine, very painful. And why do we care about this is because a single fracture in Duchenne muscular dystrophy, we know predicts further fractures. So not only are they painful, but they end up leading off with a cascade of more fractures, can lead to more deformities, and in some situations, even a life-threatening condition called fat embolism syndrome. So we want to find these fractures as clinicians. We want to find these fractures as soon as possible because there are medications that we can start to help maintain bone strength and try to prevent fractures. However, if we don't find them early enough, so here you have a spine x-ray of the vertebrae. There were only mild changes here at the spine. This child was not treated in time and ended up developing much more severe fractures, which are much harder to stop the process once it gets going like that. So what can we do? We can do bone density scans to try and measure how strong the bones are, but the bone density scans aren't really sensitive enough to find the earliest signs of fracture. But an individual's bone strength or bone density is actually highly determined by its genetics. So 60 to 80% of someone's bone strength can be determined by all of the genes in their body. And so we can try and couple that, if we can measure it properly, and couple that with environmental factors to try and get a better predictive risk of who will develop fractures early and who will develop more severe fractures. So we can accumulate all of these small changes into something that's called a polygenic risk score, specifically for osteoporosis. And this can be measured by taking a blood test or even a saliva swab. So what is a polygenic risk score? Well, imagine very small changes in small parts of different genes. And so what happens is that imagine that it is like little puzzle pieces, and individually they're not really important, but the more that you can tabulate them, the more that you can add them up, you can kind of get a bigger picture of what's going on. And so this is what we can do when we can take somebody's blood and extract all of their DNA and then measure the amount of how much each single little snippet of gene contributes to the overall picture. And we can then kind of classify who is more at risk and who is less at risk. And what are our key objectives by doing this? Well, we're trying to determine, does the polygenic risk score for osteoporosis predict low bone density in fractures in children and teens with Duchenne muscular dystrophy? And we'll try and couple that with other risk factors that we know as well, like ancestry, age, sex, the stage at puberty, medications, and other conditions, or deficiencies in calcium and vitamin D intake. What do we expect to achieve through all this? Well, we know that the PRS score has been shown to be linked with fractures in adults at risk for osteoporosis. So we hope to show that there is an association, or there is a link in the risk of fractures and bone fragility in children with Duchenne muscular dystrophy. So we're trying actually to be better at predicting who will get the osteoporosis and when. And by combining that with conventional risk factors, hopefully will improve screening and in-store treatment. So what could be the impact of the study? Well, earlier detection. Imagine having a smoke alarm instead of actually waiting for the fire to happen. It's also a more personalized medical approach. So far, we're using just general protocols and screening everybody and treating everybody the same way. But this would lead to better screening of people who are more at risk, less tests, less screening, less hospital visits for those less at risk. It's actually quite cost effective, only costing $80 Canadian for a single sample, easily available with, like I said, a saliva swab or a blood test. And it would really help doctors and clinicians improve how they treat osteoporosis, treating the ones most at risk earlier, and maybe decreasing the amount of tests we need to do in the ones that are less at risk. So earlier detection, hopefully fewer fractures, fewer hospital visits, and hopefully it could contribute to improved quality of life as well. So thank you so much to Muscular Dystrophy Canada for this grant. I want to say, and especially all of their donors. A special thanks to Dr. Despina Manusaki, who is a co-applicant with me, and Dr. Agath Rio, who is a fellow on this project. And also many thanks to the entire neuromuscular and bone health team at Saint-Justine, and especially to all the teenagers and children with Duchenne muscular dystrophy that I have the privilege of following. Thank you.
Congratulations on your work, and we look forward to the outcomes of an important project. Up next, we have Dr. Claudia Cote from working together with Dr. Cynthia Gagnon, and this is another project that we're getting to support in Friedreich's ataxia as well as myotonic dystrophy. Over to you.
Yeah, hi everyone. I'm sorry I don't have a visual support, but I'm really proud to present our research project and to be co-principal investigator on this project with Professor Cynthia Gagnon. Our project focuses on improving how we assess swallowing difficulties in people living with myotonic dystrophy type 1 or DM1 from the patient's perspective. And we are happy to include also people with Friedreich's ataxia. And this project is important because swallowing difficulties is a condition experienced by many patients with neuromuscular disorder. It can be up to 80% of people with DM1. It affects nutrition and also increases the risk of aspiration pneumonia, which is the leading cause of death in DM1. And despite how common and serious this problem is, we still don't have a clear, consistent way to ask people about their swallowing or to measure how much it affects their daily life. And without that, it's hard to track symptoms properly, guide care, or test treatments in clinical trials. So that's the gap we want to address by this project. And we aim to select, adapt, and validate a patient-reported outcome measure, a questionnaire that reflects the real-life experience of swallowing difficulties in DM1 and Friedreich's ataxia. And this means creating a questionnaire that is clear, relevant, and tailored, not just scientifically accurate, but also meaningful to the people who live with these conditions. And we are starting by working with an expert panel to identify the most important swallowing symptoms and which ones are likely to change over time. We'll also conduct interviews with people living with DM1, with Friedreich's ataxia and their relatives, both in French and English, to make sure that the questionnaire makes sense and reflects their experience. And finally, we'll run a pilot study with 60 participants to test whether the questionnaire really measures what it is supposed to. And with the right questionnaire, we'll be able to detect swallowing problems more effectively and follow their progression more precisely. Ultimately, this is about improving how we listen to patients and how turn their voices into better care. And although we're focusing on DM1 and Friedreich ataxia, this work could also help other neuromuscular diseases as well, because dysphagia is common in many of them. And I'd like to thank DMC for supporting this project and all the collaborators. We have collaborators from the Netherlands, from France, and the United States and Canada. And well, thank you to all the community.
Wonderful. Congratulations again, Dr. Cote. Up next, and definitely not last, but Dr. Jodi Warman-Chardon is going to wrap up this section, round out our section on clinical research. So over to you.
That's great. Can you see my screen?
Not yet, but I do see it starting, and I do see it now. Over to you.
Okay, that's great. And you can see it in presentation mode? Yeah, great.
Not yet, but did you put, there we go.
Okay. So thanks everyone. I think I'm the last talk, so I will not take too long, but just wanted to thank MDC for this grant. So this grant is focused on biomarkers in inclusion body myositis, looking at imaging and proteomics. So inclusion body myositis is an acquired autoimmune disorders that particularly causes weakness in the hands and in the quadriceps, and specifically trouble swallowing. So there's about 2,000 people living with IBM in Canada. And the muscles that are mostly affected are swallowing, and people often describe trouble choking and having to spit up their food, difficulty using their hands, and getting out of chairs. Now, IBM is particularly difficult to diagnose because there are many similar diseases, especially early on. There's over 400 different muscular dystrophies and autoimmune disorders now that resemble IBM early in the course. And muscle biopsy can be patchy and miss the important signs on IBM. And so, as with many slowly progressive neuromuscular disorders, these are really difficult to assess in trials. So most of our clinical exam markers, and you can see the theme through the clinical talks, are really just not sensitive enough to assess disease progression or treatment response. And so the biomarkers, a way to better assess disease, will help better diagnose people with inclusion body myositis, assess disease progression, and address treatment response for trials. And when I see patients in clinic, I say it's not just about finding the drug, which is critically important, but it's about proving that the drug works and convincing the government and insurers to pay for it, because a lot of these medications are so expensive. And so if you look at the natural, disease progression, we hope that medications will slow progression, stop progression, or recover function. And so it's critical to have biomarkers that are sensitive enough to show these changes. And so the objective of this study is to, IBM2 is to find these blood and imaging biomarkers to better diagnose people living with IBM and assess treatment disease progression. And so we've got a three-pronged approach. And the first of all is to look at validated measures and compare validated measures in IBM and strength testing, biopsy results, patient-reported questionnaires, swallowing studies. The second aspect is to look at muscle imaging. And so when we look at muscle imaging, so this is at the level of the calf. This is just a standard MRI. And if you look at the image on the left, you can see that the gray is good. So those are healthy muscles with sort of normal subcutaneous or the fat tissue that we have underneath our skin. And if you look to the right, you can see that each muscle is individually assessed. In IBM, what happens in many different neuromuscular disease, there's a very specific pattern of muscle involvement. So you can see the white or the fatty involvement of the muscle as the disease progresses. So we can look at each single muscle along the length of the muscle and assess disease progression. And so we can look at the whole body at specific muscles along the length and compare the first time that they have a scan and then follow the scans over time and look at the disease progression. And this is a bit busy, but compared to healthy controls, we can look at both the fat fraction and the amount of water and edema, and to look at these sentinel or early disease changes in muscles. But not everyone can have an MRI, and some people are claustrophobic, or if you're living in remote areas, muscle MRI is very objective, but it's not practical for every patient. And so what we're pairing our imaging with is certain blood proteins or what's called proteomics. So we're looking at 11,000 different proteins in a very well-validated test and following our patients over time. And so if you think starting early in the disease and then as the disease progresses over time, how the muscles secrete these different proteins in the blood, and then we're looking at the blood and identifying which proteins are higher versus lower throughout the disease, and then compared to healthy controls. And so what we're hoping to do for this study is to compare the clinical data, these very specific muscle MRI imaging. We're one of the only centers that are able to offer it, that looks at specific fat fraction and water biomarkers, and then look at the protein expression. And we're looking at this over time with regular analysis as well as machine learning. So this artificial intelligence, as some of my colleagues are also doing, to see if we're able to better predict who is going to progress and who might better respond to potential treatments. And so, the impact of this study is to look at the fat fraction and edema in muscles and assess these as biomarkers for clinical trials, looking at these very specialized proteomic blood biomarkers for trials. And this is being used for inclusion body myositis, but it's a model that can be used for many neuromuscular disease. And I would specifically like to thank the very kind patients who I have in clinic, and some of you may be online, and thank you sincerely for everyone that's contributed preliminary samples, because that's critical to help the research and moving forward. I'd like to thank my colleagues in radiology and bioinformatics with their collaboration with this study, and of course Muscular Dystrophy Canada for the funding. And I can speak to everybody in the panel about how appreciative we are for this funding to move the research forward to help people living with muscle disease. Thank you very much.
Thank you very much, and congratulations once again. Before we go to our fireside chat, what I'd like to do is I see a number of comments and questions in the chat section, so I welcome anyone else who would like to put those questions in. I'll go ahead and read the first comment, which is from a parent with a child with Emery-Dreifuss. So Dr. Curry, this is for you. It's more of a comment to say it took 6 years for their child to be diagnosed. So anything in terms of improving that time to treatment or time to diagnosis is welcomed. And so that really resonated your project with them. So that's just a comment that I wanted to share. And we also have 2 questions that are similar, but first I'll direct it to you, Dr. Barnett-Tapia, which is to say, we heard on today's webinar about OPMD and its impact on speech and swallowing. And while you did mention that your work would be for myasthenia gravis, do you anticipate that collaborating with those who work in OPMD, you could then extend this work to patients affected by OPMD?
Absolutely. And I was thinking about that when I was listening to the other talks. It's really this platform would help any individual affected by any neuromuscular disorder that causes speech, swallowing, chewing problems. So we may need to tweak it for other diseases that present a little bit differently and also for ages. But for sure, this is the first step on expanding to other neuromuscular diseases.
And then a similar question for you, Dr. Fiscaletti, which is in terms of osteoporosis is present in other neuromuscular conditions, and they see a high value in the work that you're doing outside of Duchenne muscular dystrophy. Any plans in the future or ways that this work can be applied to others or that score?
Yes, absolutely. So we'll start with Duchenne muscular dystrophy, but then of course to apply it to more heterogeneous or varied diseases such as spinal muscular atrophy and other types of muscular dystrophy. Absolutely.
Dr. Warman, a question for you, which is whether you anticipate biomarkers to look different throughout disease progression based on the age of the patient.
That's a great question. So that is the importance, and I think for many of the panelists, about assessing the disease biomarkers over time. So, particularly for genetic neuromuscular disease, we know that people are born with this, but you might not have any signs until later on. So many of the studies are actually looking at the pre-symptomatic patients or the patients that might have tested positive but don't have any clinical signs, and follow over the disease course. And what, some of the blood biomarkers we know, we've got studies in oculopharyngeal muscular dystrophy, which is popular for this panel. And actually, this was a study also funded by Muscular Dystrophy Canada in the past, that our pre-symptomatic patients didn't show any changes on the blood biomarkers early on in the disease. And it was just later on that we saw a change. So it was really important that we pick the right tissue to be able to show a difference. The MRI showed changes earlier than the blood did. So that's a great point. And it's different for every different disease. There's over 600 different genetic neuromuscular disease and at least 100 plus autoimmune diseases. And so each one will need different biomarkers throughout the course of the disease.
Excellent. If there are any other questions, please feel free to put them into the chat and I can definitely address them closer to the end of the webinar. I would say a lot of the other comments are really just excitement that this work is happening and already volunteering to participate, and we're happy to support you with recruitment when the time is right. And of course, with, as Dr. Parks mentioned, knowledge translation, many are really eager to hear about the findings of this work. So up next, we are really pleased to have well, you do have an honorary doctorate, Danielle, so that is appropriate. Danielle Campo-McLeod, who's one of our patient research partners. We're really pleased to have Danielle as our national ambassador. She's personally affected by a neuromuscular condition, and I'll go ahead and stop sharing our screen so we can see you. And we're going to have a fireside chat with one of the first questions to say, as someone who's personally impacted, you've now heard from several different researchers. What are your thoughts on the research that you heard about today? How did it make you feel?
Well, I first off want to just say thank you. I think I'm just sitting here filled with gratitude. And I know that, for all of these amazing research projects that we hear, I know the hours of dedication that has gone into, trying and trying again and trying again. And As someone who lives with a neuromuscular disorder and as a parent of 2 young children, I'm just filled with gratitude, but I'm also filled with hope. And I can feel your passion coming through the screen. And I think for all of us watching that are affected by a neuromuscular disorder, that means so much. When you receive those words that it's you or that it's your child, you suddenly feel so alone on your own little island. And what I felt tonight was just seeing those bridges being built and us all being together as one to move this research and amazing clinicians to help walk alongside us through this. So I'm just filled with excitement and gratitude for all of the hours that you have put into all of this work.
Wonderful. Thanks so much, Danielle. And as everyone on this call knows, MDC's mission is to find cures and this work gets to us closer to finding those cures. In your view, Danielle, how do donations, whether big or small, help to bring that mission to life?
Well, I think it's so important and we're seeing it happen. I think, you know, every dollar matters. And I look at what has been donated and what has gone into the research and all of the work that Muscular Dystrophy Canada does for people living with neuromuscular disorders. So I never want someone to feel like, you know, a dollar is not significant, because we're seeing, as one of our doctors said, when you put those puzzle pieces together, how important they are. And it's the same with the dollars. When we put these dollars together, look at what we can do and look at what we are doing. So I think it's so important to help us achieve this mission.
Danielle, when you reflect on your own journey with the neuromuscular condition, now as a parent of children with neuromuscular conditions, how do you think research has played a role in your journey so far?
Well, I've walked closely alongside with many on these calls, and I think it's so important. For me, I was diagnosed with a different neuromuscular disorder at the age of 2, and following, you know, mapping that, I just didn't quite fit that box, and we kept looking. And then as the research moved on and we could test more genes and we could get, you know, different tests and have better understanding, I was able to then get my final diagnosis of spinal muscular atrophy, as well as being able to hear the words that it's not if there will be a treatment, it's when there will be a treatment. And when you're a parent and you hear that for your children, you hold on to whatever string of hope you can. And so for me, it's been really important. It's so important to have that title. And I know that that might sound funny, but when you're walking alongside, I think Dr. Park said it best, we need to know, what does it look like in the future? Well, you have questions, right? As I celebrated my 40th birthday this year, I was so excited for that, but then also like, wait a minute, that needle's going further on the other side. What is that going to look like for me? How do we prepare for this, as well as for my children? How do we prepare? And being able to have those answers, or to have the ability to say, you know what, we don't know right now, but this is what's happening, so we do have those answers. So for me, it's been a long journey, but really exciting to see that those questions are getting the answers and that hope continues to ignite and be there.
For many people, research can feel like a big black box. It's really sometimes distant or complex. And I'd like to applaud everyone on the call today for really doing these flash talks, such robust, comprehensive proposals submitted in just 3 to 5 minutes sharing it. But Danielle, how would you explain the real life impact of research for someone who's never been really touched by this kind of work?
Yeah, I think first of all, I have to thank Muscular Dystrophy Canada, because the way that we walk alongside people in that knowledge translation to help, it sometimes can feel like we're speaking different languages. So I think that's so important. I think for everybody to just take a deep breath and to know that we have the incredible researchers and clinicians behind us, and that not to be afraid to ask those questions, right? To reach out, to connect with Muscular Dystrophy Canada, but to connect with your doctors, ask those questions that you may have, and take the time to say, you know, research is exciting, but you said it best, Ramira, I can sometimes feel like, where do I turn? Or I have to read this, or have to read this article, and I can't even read the words in the title. And so it can feel overwhelming, right? So just to take that minute to kind of take that deep breath and know that there's such an incredible team behind you, and that as a patient and a person living with and affected, you have the right to ask those questions and to ask your questions.
Wonderful. And the last question I had is, you know, it was really powerful when Dr. Parks presented the example with spinal muscular atrophy and how so much has changed over the last just even in the past 5 years alone, in the last decade alone, 3 disease-modifying therapies, newborn screening across every province and territory. Looking ahead, what gives you hope? When it comes to research, when it comes to your journey with the neuromuscular condition, as you work with and meet with clients, donors, firefighters at Muscular Dystrophy Canada?
I think what gives me the most hope is that we have this incredible community behind us. When I said that passion is coming through the screen, I speak to the researchers and the clinicians when, it's your sister, it's yourself, it's your children, it's your mother, your father, your brother, that these are the faces. So you may see a mouse on the screen, but those are the people that are walking alongside, living through this. And what gives me hope is that there's that community behind you, that we know that look how far we've come. And I think SMA is an amazing example. And to know that, yes, it takes time and it takes thousands of people, but I thank you for the people that this is their passion. And we know this isn't 9 to 5. I know they wake up at 3 o'clock in the morning thinking about this. I know that it's, 7 days a week that they're watching their kids on the soccer field, and then poof, that research thing pops into their mind. And we feel it as the people living with the neuromuscular disorders. So I want to say thank you for all of the work that they do. It does provide us hope. And also to the people that are living with it, that walk alongside it, it helps you push through those struggles when you know there is a community behind you, supporting you and actively looking into this. So it just gives me hope to know that Muscular Dystrophy Canada is there for you and helping to invest so that we do one day say, we have found that cure, we have found that treatment for all 500, 600, however many neuromuscular disorders and autoimmune disorders that there are. And so I'm hopeful. I see the work, I see the amazing changes, and it makes me very excited. As a parent of just, a 6-year-old and a 3-year-old, I'm filled with hope. And that feels pretty incredible to say that, yes, they have spinal muscular atrophy, but yes, we have a lot of hope.
Wonderful. Thanks so much, Danielle. Thank you for taking the time this evening. On a final note, I welcome our CEO, Stacey Lintern, for some closing remarks.
Thank you, Homira. Good afternoon, good evening, everyone, and thank you so much for joining us today for Moving Research Forward, the result of the 2025 Neuromuscular Research Competition. Today is truly about celebration. Danielle expressed that so well, celebrating innovation, collaboration, and hope for the future. First, I would like to extend a heartfelt thank you to all the researchers who submitted proposals for this year's competition. Your dedication, your passion for advancing neuromuscular research is truly inspiring. Congratulations to all our successful translational and clinical grant recipients. Thank you for your time today and sharing your information about your research projects. We're excited to see how your groundbreaking work will transform the lives across Canada and beyond. A sincere thank you to the reviewers as well. Your expertise, your thoughtful analysis, your commitment to fairness, ensuring that only the highest quality of research moves forward. And none of this would be possible without the generous individual donors, supporters, and incredible Canadian firefighters who tirelessly champion the neuromuscular cause. Because Muscular Dystrophy Canada does not receive any government funding for research, your support quite literally empowers innovation and progress. I also want to thank and recognize our research team led by Dr. Homira Osman at Muscular Dystrophy Canada and Dr. Robin Parks, the chair of Muscular Dystrophy Canada's Medical Scientific Advisory Committee. Your leadership, diligence, and compassion guided the entire grant competition process this year, and your efforts have a profound impact on the neuromuscular community. A heartfelt thank you to Danielle, Muscular Dystrophy Canada's National Ambassador, for your ongoing direction, insight, and expertise. And of course, we are so grateful for the Canadian neuromuscular community who work in partnership with us to continue to search for cures, better treatments, standards of care, better and the earliest diagnosis, which is so critical. And it's so exciting to hear all of the work that's helping move the needle with the earlier and better diagnosing process. As we celebrate today's successes, I'd like to invite each and every one of you to continue this momentum by joining us at a local walk and roll event in your community. It's a wonderful way to support research, connect with families, and raise vital funds. You can register for Walk and Roll or make a donation by visiting Walk Roll MDC. A link will be shared with you after the webinar. If you have any questions or comments, or you'd like to learn more about Muscular Dystrophy Canada or explore ways you can support our research efforts, we encourage you to reach out to Muscular Dystrophy Canada's research hotline, We'll share this link, contact information for the research hotline after the webinar as well. We're here to help, and we would love to hear from you, and we would be grateful for your support. Together, we are breaking down barriers and moving research forward on behalf of the thousands of Canadians affected by neuromuscular disorders. Thank you. We are so grateful.
Thanks so much, Stacey. Thank you again to all the recipients of this competition. We're really eager to look forward to your research outcomes, and we're here to support along the way. Thank you again to our donors and those who are participating in today's webinar. Thank you and have a good rest of your day.
Thank you.
Thanks, everyone.